my name is Dustine Reich, and today I will be presenting on the experiment I performed entitled The Locomotive Defects in Drosophila model of Huntington's disease. So I first would like to start with the background on Huntington's disease. It's an autosomal dominant disorder that causes progressive neuronal death, and it's characterized by uncontrollable movements, behavioral changes, dementia, and eventually death. It's caused by mutations in the Huntington gene, which contains glutamine repeats, which is represented by the letter Q. It has previously been found by Caterino and other researchers that the number of glutamine repeats determines whether an individual will develop Huntington's disease and its severity. For reference, in humans, 36 glutamine repeats is a threshold for Huntington's disease, while over 40 repeats will always result in Huntington's disease development. So the objective of this experiment was to use Drosophila as a model organism to study the relationship between glutamine repeats and Huntington's disease associated locomotive defects. So we use flies expressing Q16 repeats and Q128 repeats. So flies expressing Q16 repeats will not develop Huntington's disease, and therefore it's predicted that they will have comparatively better locomotive abilities. Flies expressing Q128 repeats will develop Huntington's disease, and therefore it's predicted that they will have locomotive defects. This experiment was performed by crossing a male fly line with two different female fly lines to produce flies expressing Q16 and Q128 in neuronal tissue. A startle-induced geotaxis assay was performed using 10 of each to determine how the polyQ repeats affected their inherent negative geotax response. This response is basically their inherent response to always crawl upwards. The assay measured how many flies reached the 50 milliliter mark on a 100 milliliter graduated cylinder within 30 seconds. This here is an outline of the trials. So there were 3 different trials. Each trial, the flies were shaken to the bottom of the cylinder, a stopwatch was started, and after 30 seconds, the number of flies to reach the 50 milliliter mark was recorded. So this occurred 3 times. There was a break of 2 minutes, and then these 3 trials were performed twice more. So overall, there were 3 different trial sets performed for both the Q16 and Q128 flies. So the result was that on average, 44% of the Q16 flies reached the 50 milliliter mark within the 30-second time frame, while only 27% of the Q128 flies were successful. So overall, the Q16 flies outperformed the Q128 flies by 17% on the negative geotaxis assay. I supply this picture here so you have a better idea of what the actual test looked like. This is the 100 milliliter cylinder, and you can see this orange mark is the 50 milliliter mark, which the flies were counted to see how many reached them after the 30-second time frame. This graph here shows the comparison of these 2 different flies' ability to complete the negative geotaxis assay. This blue column represents the Q16 flies, and this orange column represents the Q128 flies. So a taller column corresponds to a greater average percentage of successful flies, and therefore a quicker negative geotaxis response. From this graph, it's really visually apparent that the Q16 flies were more successful than the Q128 flies at completing the assay. So the discussion of the results is basically that the data indicates that the flies expressing Q128 repeats had greater locomotive defects than the flies expressing Q16 as a result of Huntington's disease development, as was predicted. So the Q128 flies were less successful because they had a reduced inherent negative geotaxis response time, indicative of locomotive defects as a result of Huntington's disease. So these findings further demonstrate the relationship between glutamine aggregation and Huntington's disease associated locomotive defects. Further experimentation will include repeating this assay as well as doing another test to see how age affects the Q128 flies, and it's predicted that older age will result in greater locomotive defects in the Q128 flies. So as shown through this experiment, Drosophila can be used as a model organism to effectively study Huntington's disease. These findings further add to the scientific community overall understanding of the disease to better aid in the discovery of future therapies. I would finally like to acknowledge the Department of Biology at Brandeis for supplying the equipment and space for this experiment, as well as Joyce Riggle and Kendrick Aruno for always providing support and essential on-site instruction. Thank you for watching, and I hope you enjoyed.